Good afternoon. So today we will learn regarding the one of the type of cytoskeletal structure, that is microfilaments. Okay, so as you can see it on the screen, microfilaments are the thinnest class of cytoskeletal fibers, which are solid rods of globular protein called as actin. I can remind you that there are other different type of cytoskeletal structure named as microtubules and intermediate filaments. Out of the three we are discussing today only one, that is microfilaments. So microfilaments are the thinnest class of cytoskeletal fibers, which are solid rods of globular protein, that is nothing but actin. What is actually an actin? I'll explain it in detail regarding about it in the coming slide. So an actin microfilament consists of a twisted double chain of actin subunits. As the DNA will be intertwined with each other, twisted rods of actin monomers, it is nothing but a actin microfilament. Okay, so such two microfilaments are intertwined with one another to give rise to one microfilament. Okay, so microfilaments are designed to resist tension. That means the tension, there will be tension in the cell where while doing its function in the animal cell, particularly as well as in plant cell, to resist that tension, particularly the microfilaments which are made up of actin protein are involved. Okay, then with other proteins, they form a three-dimensional network just inside the plasma membrane. Okay, fine. You can see the structure of the microfilaments here. The details as well. Structure to intertwined strands of actin. This is a particular cell from where a part is highlighted into the microscopic structure and in the animated structure. So this is entirely a actin microfilament. Okay, and in which out of it this one globular part is nothing but an actin subunit, and the thickness of this particular filament is 7 nanometer. Okay, the main functions of the actin, what we call microfilament, is maintenance of cell shape, changes in cell shape, muscle contraction, cytoplasmic streaming, which is also called as cyclosis. Okay, then cell motility, that means the movement of the cell, not the movement, I mean not the cell will be showing the movement from one place to the place, but the cell's movement by remaining at its own place. The dynamic structure of the cell, it will be maintained by the what we can call this particular microfilament. That is actually the meaning of cell motility. Then as well as they also help in cell division. Okay. In the electron microscopic photography, the microfilament will be looking like this. This is microvilli, the microfilaments, which is also called as actin filaments, and intermediate filaments, which are larger to this particular thing. Okay, you can see the footnote of this particular photograph, the shape of the microvilli in intestinal cell. So this is the photograph of a single intestinal cell, a part of the single intestinal cell which is showing the microfilaments. Okay, anchored to a intermediate filaments, a network of intermediate filaments. Okay, then in muscle cells, thousands of actin filaments are arranged parallel to one another. Okay, that means in one single muscle fiber, thousands of actin filaments are what we can call arranged parallel to one another. Arranged parallel to one another to form to give rise to single muscle fiber. Such many muscle fiber will give rise to single muscle cell. Okay, so thicker filaments composed of motor protein myosin interdigitate with thinner actin fibers. That means myosin is the muscle protein which is more in number, more in concentration, and which is making thick bands of the muscle. And thinner will be the actin fibers, which will be consisting of actin microfilaments. Okay, so myosin, as you can see it on the screen, myosin molecules walk along the actin filament, pulling stacks of actin fibers together and shortening the cell. That is how they undergo contraction and relaxation. Okay, in other cell, for example, this is a amoeboid movement using in the amoeba organism, microscopic organism. In other cells, these actin myosin aggregates are less organized but still cause localized contraction. They are not organized in lower organism, but they can cause local movement. That means a movement in a part of a cell. Okay, a contracting belt of microfilaments divides the cytoplasm of animal cells during the cell division. Okay, fine. Then localized contraction also drives amoeboid movement. Pseudopodia cellular extensions extend and contract through the reversible assembly and contraction of actin subunits into microfilaments. Okay, you can see it in the figure here. The movement of the microfilament will be giving rise to a cyclosis in the cytoplasm of the amoeba, and that will give rise to a pseudopodia like structure that will help this particular organism in locomotion. Thank you.